Welcome everybody to another episode of the Bioinformatics Chat. I'm going to be your host today, Jacob Schweppe, and today I have with me Dr. Devin Schweppe. Dr. Devin Schweppe earned his PhD working with Dr. Scott Gerber at Dartmouth Medical School. After that, he went on to do postdoctoral work at the University of Washington and then at Harvard Medical School. Recently, he has returned to the University of Washington as an assistant professor of genome science. Thank you for joining us.
Thanks for having me.
So I actually met you last year at the Genome Science Retreat. In one of those instances, you were schooling me about the polycomb repressive complex. You thought that my ideas on how that worked on the genome were outdated or wrong.
Sounds just like me, yeah, totally.
And then we also competed against each other in The Price is Right.
That was a brutal competition, yeah.
Yeah, if I recall correctly, then in the finals we had to guess the price of a piece of instrumentation, a piece of equipment that was in your lab.
Going to be, yeah.
Going to be in your lab,
I think I missed it by 10%. So close.
You were very close, but you were very slightly above the actual price. And so me, who was very wrong, but underpriced, still managed to win.
Classic Price is Right rules.
Right. Okay. So now you've been a professor at the university. You started your position this fall, right?
I started July 1.
Wow. What a great time to be starting a new professorship position.
Oh yeah, real no stress period in the world. So
what's it been like starting a professorship during this time?
Moderately chaotic in some senses. Obviously hits to logistical networks, and shipping has been a challenge for getting instrumentation here and getting things set up. But I would say the one great part about the scientific community in general, particularly genome science, is that people are not only quite kind and outgoing and helpful, but also very supportive, even and especially in times like this. So it provides a great resource to kind of get those things that maybe aren't shipping on time, those reagents that we don't necessarily have on hand because we're a new lab, that we can borrow from the likes of Mike McCoss or Judith Yen upstairs. So.
You can just go invade their labs,
Of course, we will tell them first.
Leave IOUs.
But yeah, it's been a fantastic kind of resource. And of course, as you know, Bill Noble, who's transitioning to become the chair of the department in the interim phase, has also been a great resource in terms of talking to him about setting up a lab in the department. So with the chaos, there still comes a great support network here and also abroad, talking to colleagues and folks going through the same thing across the country, around the globe. So.
Right. I don't envy you that for, computational folk like me, though, of course, for everybody, there are struggles. For computational folk, I recently transitioned from being a postdoc at the University of Washington to being a postdoc down at Stanford. And so for me, the transition was as simple as logging out of my Bash terminal and, that I was working on before and opening up a new Bash terminal. And suddenly I was working at Stanford using their computational resources. For you had these huge, as you mentioned, these huge logistical struggles that you had to literally purchase equipment and stuff. Have you been able to get that, or is it still significantly delayed?
I would say it's right now about a 50-50. So a lot of the equipment we've gotten shipped. We've had some really great interactions with some of the sales reps, and they've with knowledge of the current logistics situation helped us out a lot trying to get equipment here as fast as possible. So basically everything in the pipeline right now for us is here. The one thing we're waiting on is the most important thing, unfortunately, which is the mass spec to arrive. So that should be here in the next couple of weeks, and then I think we should have just about everything to kind of be up and running fully. So we're really excited about that.
I see. So now you just in the middle of the night, you secretly run over to someone else's lab and use their mass spec.
Fortunately, we've had some very generous offers from Judith and Mike, and in terms of potentially running samples if we really needed to. And also, the big resource for us is Priska and Jimmy down in the UW-PR, so the proteomics resource or core here, where we can do emergency sample analysis if possible in the next couple of weeks.
That's awesome. Have you, other than trying to get equipment into your lab and functioning, have you noticed any type of other, How is the experience of starting a professorship going?
I think it's one of these things that's kind of hard to describe and hard to compare because everyone goes through it in a manner the same. But of course starting now, you have COVID. Starting a few years back, there were other issues, but, I would say as best as I can tell, it's going smoothly. I hired a research scientist. We're effectively right now looking to hire some postdocs and hopefully going to get some rotation students coming through in the next couple months. And all of that seems to be kind of flowing together. It's now kind of the pursuit of funding and making sure we can get these people funded and sustain kind of the lab and lab infrastructure. But yeah, I guess the question of how's it going for me always drives the idea of how do you compare yourself to how it's gone for other people and all the kind of background things that come into that and complete that comparison.
I mean, it sounds like the classic thing in academia where everyone is on a unique trajectory, but everyone is also comparing themselves to everybody else in an unfair manner.
100%.
I mean, why don't you have as many papers out as Bill Noble?
I know, right? Why don't I have as much funding as Bill Noble? I mean, he's only been here a few years, right? So I should just be there.
Yeah, exactly. So you were mentioning that you were bringing on new people. Are you currently hiring? Are you looking for people to join your lab?
Yeah, of course. So we're really excited. So we finally just got approval for a postdoctoral position and hoping to fill one of those now and also looking for some graduate students. So we've got a bunch of projects and we're trying to kind of get as many hands on board to increase our bandwidth to start getting those projects through and start publishing papers and getting those grants in. So yeah, I'm really excited for trying to get as many people as possible in. So, but of course then there's the COVID situation and making sure people are safe when they come here and all that stuff.
Right, exactly. If people are interested in joining your lab, how should they get in touch?
I would say the easiest way is just go to schweppelab.org. You can find contact information and reach out to me that way and kind of take a look at projects that we have going on and that we'll be taking in the future. So.
Awesome. So let's get into it. The work that you do centers around this field of proteomics. Can you describe to us what proteomics is?
Sure. Proteomics in general is the analysis of the proteome, so the compendium of proteins in a given living system. So essentially, what we're trying to do in the early days of proteomics was simply to establish what proteins existed at a given period of time. You can imagine doing an immunoprecipitation and trying to identify all the proteins that came down with that. Given bait protein or antibody, giving you an idea of protein interactions. And over the last few years, the field is moving more and more towards quantitative aspects. So really trying to understand, can we compare across multiple different conditions or cellular states or across time and drug dependency to try and understand how the full set of proteins, so somewhere in the order of 10 to 15,000 proteins in a given cell or tissue for humans or mouse, how those change throughout those conditions or across time. That quantitative aspect has lended a lot of power to essentially how we can understand biological systems, particularly these highly dynamic biological systems with many, many moving parts, which in this case, for our type of analysis, is all the proteins.
That's awesome. So you're saying that you're trying to distinguish between the composition of the, 10 or 15,000 proteins within cells in different conditions. Can you give like an example of why you might want to do that?
Yeah. So, one just general example, right, is trying to understand, what are the effects of treating with a certain drug, right? So, often we know that drugs are designed to be highly specific, but often have off-target effects, say. So one thing we can do is actually look for how do cells respond to a drug treatment and look to see if the drug is meant to, say, degrade one protein and one protein only. We can look to see are other proteins being degraded upon drug treatment. And particularly what we can look to see is if we treat with multiple different concentrations, where do those off-target effects begin? So that's a relatively simplistic version of it. you can imagine other things that we've kind of tackled over the course of the work that I've done in the various different labs. We've also looked at trying to understand how post-translational modifications shift throughout the cell cycle, how protein interactions change or are established in mitochondria and the general cell during essentially host-pathogen interactions. It's basically if proteins are involved in a cellular system, we can go after them. And I think that's what's really appealing about proteomics in general.
So you're saying that there are analysis methods out there that allow you to not just quantify, the broad spectrum of over 10,000 proteins, but that they are sensitive enough in order to identify changes in concentrations of one?
Yeah, totally. And I think that's one of the really fun aspects of kind of developing these methods is over the years that sensitivity threshold for detecting just that one has shifted more and more. So we can go after proteins even in very high dynamic range backgrounds. So you imagine there's a lot more tubulin than there are transcription factors. So even in the case of trying to understand complex biology across this kind of large dynamic range, the current state of proteomics means that we can interrogate somewhere in the order of 8,000 to 10,000 proteins pretty robustly. And so what that allows us to do, is get that quantitative information about an individual protein if we're interested in that in the context of the general proteome. But also modern proteomics uses targeted approaches. So if all we're interested in is just that one protein, we can also use proteomics to go after just that one, say, transcription factor, just that one signaling molecule to try and quantify that across these conditions.
That's awesome. And so the primary tool that you use in order to do this analysis is a mass spectrometer.
Yes, sir.
I think you know my next question. Can you tell us a little bit about what a mass spectrometer does?
Yeah, so essentially what we're trying to do in a mass spectrometer is, right in the first part of the name, is trying to measure mass, right? So what we're doing in modern mass spectrometers is trying to very quickly essentially go through the full compendium of a proteome. And the vast majority right now of mass spectrometers are analyzing peptides. So rather than full proteins, we use this approach termed bottom-up proteomics. So first we digest proteins into peptides and then inject those peptides into a mass spectrometer. These peptides are ionized, and then we can measure the mass-to-charge ratio of those peptides in our mass spectrometer. And that M/Z gives us a clue as to what peptide that is, because every peptide or most peptides have a pretty distinct initial M/Z. But where we really get our sequencing information is we can take that precursor in the mass spectrometer in the gas phase and fragment that to generate a species of, or a set of ions. And these fragment ions allow us to actually sequence a given peptide. We can then take that set of fragment ions and that new potential sequence and match that back to a database. So in this case, a FASTA database to identify proteins. And we can use those identified peptides and proteins to then get quantitative information using some advanced methods such as multiplex proteomics or DA proteomics.
That sounds awesome. So the gist is that you'll have a sample and you don't know the composition of proteins in that sample, either in the form of the relative abundance of each protein or even which proteins are in the sample. And so why can't, there exist fairly commonplace DNA sequencing methods. Why can't you just use protein sequencing methods?
That's a great question. I think we would love to be able to do that, but the main problem is you can't amplify peptides, you can't amplify proteins, so it's going to be hard to be as sensitive as, say, your DNA or RNA-seq based methods. There are some high throughput single molecule technologies that are actually actively being developed that are orthogonal to mass spectrometry that try and take advantage of some of the underlying tech of those sequencing modalities, but they haven't been, I think, fully fleshed out at this point. But yeah, I mean, I think the big issue is amplification. So we need to have some analytical tool that is sensitive enough to detect what's there in that cell, without having to generate more of that species.
I see. So you're saying that generally these samples are very small, And so with DNA and RNA sequencing technologies, you just start off by amplifying it so you get a ton of it, just copying it over and over again until you have the, threshold. But, protein scientists are really letting us down here by not having a clear analog to proteins, and so you have to use a mass spectrometer.
Exactly.
Okay, awesome. So there are these terms MS1, MS2, MS3. I think I even recently saw MS4.
Oh, yeah.
I guess my first question is, why are proteomics people, why do they lack creativity?
If it's not broke, why fix it, right? So, so that whole strategy, so I kind of alluded to it, right? The strategy for taking a precursor and then fragmenting it. So essentially the MS1, MS2 idea, right, is all, the MS1 would give us precursor information. The MS2 would give us that fragment information. The, MS3 and beyond is generally when we need to do something a little bit fancier, to break up specific moieties to, do specific quantitation.
When you talk about the precursor and the fragment, can you clarify what you mean there?
Yeah, for sure. So precursor in this case means, right, so in the mass spectrometer, right, we've ionized these peptides. The precursor would be the M over Z, or the precursor mass or precursor M over Z would be the mass or M over Z for that peptide. So we initially inject those ions, we collect them in a trap, generally, or a different kind of mass analyzer, and then essentially we'll establish what the M over Z is for everything that was injected. So that might be a host of peptides. And so we then take one of those peptides, so just one peak in the MS1 or mass spectra, right? And then what we'll do is we'll actually essentially excite the ions that we've isolated from that peak and break them up, right? And so effectively what that then does is generates these fragment ions, right? And so fragment ions we observe in our MS2 spectra. And those fragment ions now are generally, based on the way we essentially excite and fragment these peptides, they cleave peptidic bonds, and that will release specific signatures of individually released amino acids. And what we can do is essentially walk along that set of fragments to then identify the specific peptide that not only had the exact mass that we observed for that precursor M over Z, but those fragment ions all match to what we would assume to be the amino acids that exist in that peptide.
Okay, that makes sense. So, I get the feeling that this is kind of like in CSI when they say enhance. That, you get MS1 and you get this very rough picture of what's in there, and you see that one thing that you're kind of interested in. You're like, enhance, do an MS2. You pull out one thing out and you get finer grained. And so MS3 is just kind of like that again.
Somewhat, yeah. So I think generally the use of MS3, just so specifically for kind of doing these peptide-based analyses, the use of MS3 is generally relegated to when we need to do something really specific, right, with that set of data. So say there's a post-translational modification and we're not getting very good fragmentation of that. So we need to isolate, again, one of those fragments. So rather than just the precursor to fragments, what we can also do then is essentially that same process again, right, to isolate one of the fragments and break that up. So if there's a post-translational modification on there, for example, we can kind of analyze that. But we can also use that for potentially quantitative methods to try and get a better idea of specific fragments, how much of those fragments actually exist in that species.
So is it fair to say that the deeper you go into this MS levels, that the more quantitative the results are?
So no, not necessarily. So right, so you can get highly quantitative information from techniques such as label-free quantitation. You can get, which relies more on kind of the chromatogram, right? So the mass spectrometer is not operating alone, right? It's operating in concert generally with the HPLC or UHPLC. So we're doing liquid chromatography up front and eluting peptides off slowly across the gradient. So we can use some information about how those peptides elute and essentially the signal we get from each individual peptide over time to get some information about the relative abundance of that peptide or species in that sample. We can also use techniques that go one step beyond and get their quantitative information from both that initial MS1 spectra and the elution of the of a precursor, as well as essentially the elution of fragment ions. And that's part of the basis for this data-independent acquisition-based strategy and quantification lab. And that's been shown by a number of labs, Michael Koss, Rudy Aebersold, et cetera, that to be a really powerful technique in terms of protein quantitation. So it's not necessarily that it's required to go deeper and deeper to get better quantification, but some methods essentially require going to that MS3 base level to get quantitation.
So what would be an example of a use case where you would need to go into MS3 levels and a use case where you wouldn't necessarily need to go into MS3 levels?
Yeah. So I think the latter would be DIA, label-free quant, right? So we don't necessarily need to go get that MS3 based quantitation. The MS3 quantitation, though, does help with this field of multiplexed quantitative proteomics. So multiplexed quantitative proteomics refers to sample multiplexing rather than essentially analyzing multiple different analytes at the same time. And it's based around this idea of isobaric, or those with the same mass tags. And so essentially what happens in this process is that peptides are all chemically barcoded with isobaric tags using NHS ester chemistry. These tags are essentially on every one of our peptides by the fact that we use trypsin to digest our proteins to peptides, which most of these peptides then have a lysine on them, but always have an N-termini with a primary amine there. And essentially what isobaric tags are is this trifunctional group that allows for binding of the tag to a peptide, and then a balancer region that effectively offsets some mass, and a reporter region. And this reporter region is filled with stable isotopes in various different ratios that are balanced again by that balancer region. And what it allows us to do is the balancer plus the reporter have the same mass no matter which barcode we're using. But as soon as we fragment our peptides, and at the same time we're actually able to fragment this tag and split the balancer and reporter region, we can actually release the reporter. And these reporters have now, the latest generation of these have 16 different reporters that can be simultaneously analyzed, which allows us to effectively tag 16 different samples and then release that reporter for this quantitative information. And so what we can do now, right, is in that MS3, what we can do is isolate some of the fragments that contain this TMT barcode. And what we can do then is, upon release of that, we release the reporter. And the reporter, because it's bound to every single one of these peptides, gives us relative quantitative information about how much of that peptide and protein exists in each one of those samples across all 16 of our samples. And so we get the best quantitative information in that type of technique when we use this MS3-based quantitation.
That sounds really cool. So let me make sure that I understand that, though.
Yeah, there's a lot there, so go.
It sounds like the main problem is that because the mass spectrometer, as the name suggests, relies on mass, that if you have multiple samples that have the same mass, you won't necessarily be mass to charge ratio, you won't necessarily be able to distinguish between them. Is that correct?
Yeah, in part, yeah. So that can generate, so right, you can imagine if we have two peptides and their precursors are very close together, that effectively if we have we isolate the region around one precursor, we might pull in some of that other precursor. And so we could potentially, one, generate what's known as chimeric spectra, right? So spectra that are essentially just made up of both of these different species. One issue, particularly with this co-isolation or co-elution of isobaric species, is that in this multiplex quantitative analysis, if we were to isolate more than one species, the ratios or relative quantitative information that we get for each one of our individual peptides. So you imagine we've isolated peptide A and peptide B in the same isolation window, and now we're going to get quantitative information that is essentially the median of both peptide A and peptide B, rather than being high specificity to just because we were interested in, say, peptide A to peptide A. So we've lost some of our quantitative dynamic range in that case, and so our accuracy has gone down. And so, yeah, like you're hinting at, what we can do is we can use MS3 to essentially go back and then do specific isolation of just a subset of the fragments generated in our MS2, right? So MS1, precursor MS2 fragments. Now we can take some of the fragments that still contain TMT in our MS2 and fragment those further in an MS3. And what we found is through that process, we're able to essentially eliminate some of this quantitative interference that we see from co-isolation of peptide A and peptide B.
Okay, I think that I'm a little bit lost at this point. I think that there were two ideas that were going on in my head for what MS3 can be, for what you were talking about with MS3. The first is that with multiplexing, it sounds like what you're saying is that you have multiple samples that you want to measure at the same time, probably for sample efficiency. If you can run 100 things at the same time, that's great. And so what you're saying is that you're barcoding each one of your things so that in this single mass spec run, you're able to deconvolve which original sample each peak came from. Is that what you were saying with this whole barcoding thing?
Yeah, so that's a great summation. So essentially, yeah, the release of those reporters gives us individual information for each one, so essentially individual quantitative information for each one of those multiplex samples.
Okay, gotcha. So what I was saying before about how it's not to disambiguate between multiple peptides that have the same mass. It's so that you can run multiple samples and thus the name multiplex.
Right. And so it runs into this issue that we got to, which is this coelution or co-isolation issue, which that is essentially this confluence of where MS3 becomes useful because of quantitative information for essentially the analysis of these multiplexed samples.
Okay, so you've explained pretty well what MS1, MS2, and MS3 are doing. Maybe you can talk a little bit about the time that it takes in order to run one of these experiments.
Yeah, so there's been a lot of work done essentially to try and optimize essentially the full workflow for doing these type of experiments, right? And so you imagine this requires two sides of things. So the first being that we need to actually prepare these samples, right? So we need to add these chemical barcodes. We need to have digested these proteins into peptides. And so that process generally takes in the order of 2 to 3 days to go from here's a cell sample. Say I have 16 different cell lines, right? And I just want to compare the relative abundance across those 16 different cell lines. And so to go from cell pellets through the process of essentially lysis and reduction and alkylation and protein digestion and then barcoding, that takes on the order of 2 to 3 days to get through. So the next phase then is essentially doing the LC, so liquid chromatography, to decomplex some of these. So we actually do some offline fractionation upstream. So that takes another maybe half day to do and to dry. And then we follow that up finally with when we get to the mass spectrometer, right? So now we can get our actual data, the things that we're actually interested in, we can start to observe. And so that process generally right now, if we were to run this method that I was hinting at, this MS3 based quantitation of multiplex samples, that process in general takes about a day and a half to actually acquire the data on the mass spectrometer. And that's kind of the state of the art for MS3 based quantitation.
I see. So it's like I was saying before, it's not just that you can run up to somebody else's lab and run a quick experiment overnight, that you'd be, running up, starting the experiment and a day and a half later, you'd have to hope that they wouldn't notice that their mass spec was running in a corner for a whole day while you were getting your results. So it sounds like sample prep takes several days, but also just running the experiment through the, like doing an MS3 takes a significant amount of time as well. Does it take less time to do an MS1 than MS3?
Yeah, so that's a great kind of hint at. So an MS1, we can imagine essentially all the ions exist, right? We don't have to generate any fragments. We don't have to do anything with the ions that have come into the mass spectrometer. So all the steps that require fragmentation in general take some amount of time. The kind of standard fragmentation scheme that's probably the most popular is this one known as collision-induced or collision-activated dissociation. And essentially that process in most mass spectrometers takes on the order of 5 to 10 milliseconds per fragmentation, right? And that doesn't necessarily sound like a lot until you realize that in a given analytical run, so say a 2-hour method, we might collect 60 or 70,000 MS2 spectra, right? So now all of a sudden you start to do the math, and now we're spending a lot of time just doing the activation before we're spending the time doing actual collection of the data in any one of our instruments. And so it will always cost a little bit of time to do an MS2. And it'll cost generally, if we're going to do an MS3 in addition, so now we have the cost of the fragmentation and the acquisition in our MS2, and we have the cost of the fragmentation and the acquisition again in our MS3. And that's also, each one of those phases is partially dependent too on what type of analyzer you're going to use. So it's a lot quicker to acquire that mass spectra in an ion trap, for example, than it is in a high resolution trap. So an FTICR or Fourier transform ion cyclotron resonance mass spectrometer or an Orbitrap mass spectrometer, which just takes longer to actually do the acquisition.
I see. So one of the reasons that you would want to multiplex is because it takes so long in order to run any one of these experiments.
Exactly.
Okay. Can you multiplex in MS1, or do you require MS3 resolution to do multiplexing?
Well, you can totally multiplex in the MS1. You just have to do stable isotope labeling, and this is like classic technique that Xiaonan and Matthias Mann kind of made famous with SILAC, right? So essentially that's dope in heavy lysine and arginine or heavy amino acids in general. And then essentially you get 2 peaks for every peptide now. So one that's heavy labeled, essentially with your heavy arginine lysine, one that's light labeled. So you can get quantitative information. The issue there was, generally with the exception of this technology that Josh Kuhn came up with, this new code idea, you could only get about 3 different samples multiplexed with SILAC. So a heavy, a medium, and a light. And so the idea was we'd like to go beyond just quantifying 3 samples, right? The more we can quantify, the faster and the higher throughput our analyses are. So we'd like to go beyond 3 to 5, 10, 15, 20, etc.
Okay, so sounds like there is significant motivation for trying to move from a singleplex mass spec experiment to a multiplex mass spec experiment. What are the challenges with doing that?
Yeah, so the main challenges are kind of twofold, I would say. So the first one is this one that I hinted at initially, which is this kind of quantitative interference that we run into. So there's this issue where if we isolate more than one species at a time, they both have TMT on them. Effectively, what we run into is this issue where we don't get accurate quantitative information for this. Right? So we move towards the median of the 2 species. Right? So that can hamper kind of the analytical workflow moving forward. The other is more in the data analysis side, right? So it's one thing to say we can do the quantitative analysis of these things and identify these peptides, but we're no longer in the days where we're sending a grad student into the basement with a pencil and a dream to try and annotate every single one of these spectra. Right? There's just too many spectra to actually get through. So we have to build up relatively robust methods in silico for doing the full data analysis pipeline on the back end. And we found that even now we're still coming up with methods to improve the sensitivity for the detection of these peptides in our mass spectrometers. Right? So it's methods for improving what we call a precursor and how we call a precursor and how accurate we can be with that. And also methods for trying to establish what are the limits of accurate quantitation that we can actually achieve. How many ions do we need to see in a mass spectrometer in order to actually call that peptide a useful quantitative set of information. So I would say those are the two main areas of active development right now. And of course, they're balanced out by, you kind of hinted at it, this idea that multiplex quantitation gives us improved throughput, as you kind of mentioned. But one thing to also mention is that because we get essentially one peptide ID that gives us quantitative information about now 16 different samples, what that allows us to do is acquire data that has fewer missing values, right? So we can get more consistent quantitation across these 16 different samples. And we can start to do just kind of some fun things in terms of designing super complex experiments that now, instead of trying to kind of assemble data through these single plexes, we can say do a full time course in one quantitative analysis in the mass spectrometer.
So why are there fewer missing values?
Yeah. So essentially, you can think of every time we get a peptide, essentially fragmentation spectra, that's going to generate, right? So that's our MS2. So we've isolated, and we have fragments for our peptide, and we've identified that peptide. We're like, we know that peptide exactly. That's exactly the peptide we know. The benefit now, right, is that when we release those reporter ions, we have quantitative information for 16 different samples, all in that same analytical run, right? It's not that we effectively are no longer missing data, because identifications can't be tied to quantitation. So a problem often in mass spectrometry is that we have slightly stochastic, or just generally stochastic, selection of ions by the mass spectrometer, right? So it's all based on intensity, and the most intense gets selected first. And sometimes the same ions aren't necessarily seen in every single pass through the analysis of a proteome. And so what that leaves us with is, if we were only to analyze one single run and then analyze the exact same sample shot again, we would not get the exact same set of peptides quantified between those two different runs.
I see. So when you say that there are fewer missing values, what you mean is that because there's inherent stochasticity in the output, that by running it a single multiplex experiment, that essentially those missing values line up across everything that you're looking at.
Yeah, that's a good way of thinking. Yeah.
Okay, so it's not that there's fewer missing values, it's that they line up across the, plexes.
Yeah.
Okay.
Essentially, when we go to remake that Venn diagram, we know that at least for those 16 samples, there's perfect overlap in the identification and quantification.
Gotcha. That makes sense. Okay, so you have a paper that came out recently where you were trying to improve the efficiency of MS3 data gathering. Do you want to give us an overview of that?
Sure. So this is a paper that recently came out in Journal of Proteome Research, and it focuses on an idea of improving kind of the acquisition efficiency for these MS3-based quantitative workflows using multiplex quantitation. So all these little nodes that we've been talking about all along have kind of built to this. So one thing, of course, that you kind of hinted at is that to acquire an MS3 just takes longer, right? So if we're going to do one MS1 and add an MS2, that takes a certain amount of time. If we add an MS3, that takes even longer. And what I've kind of mentioned is that if we move into high resolution traps to acquire this data, those require long periods of time, so 80 to 100 milliseconds to actually acquire the spectra. So one idea we had, and so Brian Erickson published some proof of principle data around this when he was part of their PE lab, looking at whether or not we could essentially eliminate the acquisition of these MS3 spectra under certain conditions. And so one thing that we actually knew going into this, right, is that when we go to do our mass spectrometric analyses, due to this stochastic nature of essentially sampling these precursor ions, we don't necessarily always generate an MS2 that matches to a given peptide that might be in our database, right? So in the typical experiment where we run a whole cell lysate, we might identify peptides for on the order of 40 to 60% of the MS2 spectra that we acquire, right? Which means we're leaving then 40 to 60% of those MS2s on the table because they don't provide relevant information about the proteins in that sample. And this could be because maybe we didn't consider a post-translational modification, or maybe there's some adduction of some set of molecules that we didn't anticipate and therefore couldn't identify. So one other aspect of this, right, is that if we're going to do this MS3-based method, so MS3 gives us better quantitative accuracy, but also requires us to spend time acquiring this more expensive, time-wise MS3-based quantitative scan. That means 40 to 60% of the MS2s don't generate a peptide that we can use, which means that 40 to 60% of those MS3s are effectively useless to us, right? And that's in the best case scenario. That's when we're running effectively a whole cell lysate-based sample. If instead what we do is we're running fractionated samples, that relative number of useful MS2s goes down to 18 to 25%, right? Which means that 75% of our MS2s aren't actually giving us useful information, which means 75% of our MS3s aren't giving us useful information. And so the idea, right, that Brian pioneered and then we started building upon was this one where if in real time, so literally as the mass spectrometer, the instrument is trying to assess what ions to select and fragment and get quantitative information for, if we could ask every time an MS2 is acquired, whether or not we should actually acquire the subsequent MS3, that would potentially let us filter and remove a lot of this spurious kind of triggering of MS3s that are useless. And so, of course, that led us to a question of how could we do this? And what we came up with is this idea of offline what we have to do, and what I kind of hinted at in terms of the bioinformatic pipeline downstream of mass spectrometry, is that we have to actually match those fragments and those spectra back to peptides and proteins, right? That's the meat and potatoes, the stuff that we're interested in. So to do that, we use this idea of database searching, right, which is essentially generating theoretical fragments and matching to those theoretical fragments for every peptide that potentially exists in a given proteome, and then using that information to essentially assign and score spectra based on peptide spectral matches, or PSMs. And so our question was, we do this process offline. It's relatively fast. Is there any way we could do this online, in real time, with database acquisition, right? So then we had to be really, really fast, right? So it's no longer we can take our time. Oh, it doesn't matter if it takes 10, 20 minutes. Doesn't matter if it takes 60 minutes to get your peptide identification. We need to get our peptide identifications on the order of 2 to 10 milliseconds so that we can actually use that information in real time with the mass spectrometric run. And so essentially what we did in this paper is we took an open source peptide database search algorithm termed COMET, which was developed here at the University of Washington by Jimmy Ang, who also developed sequest, which is one of the first and preeminent peptide spectral matching algorithms, which helped kind of establish the field of proteomics back in 1994. And we started building on this open source framework to try and do real-time search.
Yeah, so one of the reasons that I really like this paper is because rather than just having a computational problem where it's like, oh, I have a data set and I need to do something, and, usually right now I'm going to train a neural network and it takes forever, but then, there's super high accuracy or whatever. But this is actually building something into a system that needs to be working in real time because it's modulating the data collection. That with what you're facing, that the time it takes to run an MS3 is very high. And a large part of this is spent scanning useless regions of the spectrum. And so what you're trying to do is say, as this is running, can we use the information we've already gathered in order to determine, as you said, in order to determine, should I run the more expensive calculation on this region at all? And so I just thought I'm really fascinated by these applications because it involves not just theoretically being like, oh, we have a system that, can work well. It's that, this is really going to control a machine that is generating data. And so a question that I have coming from more like a machine learning background is that whenever you build these systems, there is a risk that because you're changing the data acquisition process, that you end up with bias in the underlying data. Did you look into that, and what did you find?
Yeah, totally. So it was something we were really concerned about kind of going in, because we know that a lot of what we do, again, the driver for this is always if we were to do the canonical method and then do our offline analysis, how does that compare to doing our online analysis? What do we lose? What do we gain? Where are the frictious points? What's the problem? If there are any. And so we're really concerned about essentially leaving good quantitative information on the table when we began to do these analyses, right? So what we did is actually we began with a really simple pipeline, right? So it just essentially went through and it said, here's a score. If the score is good enough, so in this case it's called a cross-correlation score. If the cross-correlation score is above a threshold, go ahead and trigger that SPS MS3, collect that data, give us quantitative information for this peptide. And we noticed that worked pretty well, right? So what we could do is just set that threshold really low. Even low thresholds essentially eliminated large numbers of these noisy or unmatched or essentially useless MS3 based acquisitions. And so what we noticed though is that we thought we could do still a little bit better. And so offline we have this full platform to actually estimate false discovery rates for our peptides. And these false discovery rates are helpful for ascertaining what we actually believe to be real peptides coming from the proteins that we believe should be in that sample. And generally offline, what we do is filter that down to a false discovery rate. So all the peptides from a given run would get filtered down to a false discovery rate of 1%. Right? And so what I did online in this paper is essentially took part of that false discovery rate based estimation online so that we could use a similar framework that allows for more flexibility and is dependent on or allows us to essentially do dimensionality reduction of a larger number of potential parameters from a given peptide spectral match instead of just relying on this one score, which made us more sensitive. But of course, then the fear is that if we filter still to a 1% FDR online in real time, maybe we messed up. Maybe the algorithm for how we're doing this estimation was just not quite trained right. Maybe it wasn't the best background training set because we're relying on previous information from this single analytical run to generate the training data for the rest of this run. So what we did is essentially just opened up the stringency, right? So instead of using a 1% false discovery rate like we would require for our offline and for establishing good databases and data sets, we essentially scaled that way up to have a false discovery rate on the order of 10 to 20%. And the idea here was that we don't necessarily mind if a few too many MS3 scans get acquired. That's not the worst. We just don't want to acquire thousands and thousands of these, right? So effectively what that allowed us to do is we're not necessarily as stringent as possible, but it allows us to try and get around this issue of severely influencing what we're collecting quantitative data on. And by that I mean essentially not collecting quantitative information when we should be or when there might be a good peptide in there.
I mean, that seems like a very reasonable idea. I think that there's a little bit of a distinction between that. There's a distinction between correctness and bias, where what you want to do is make sure you don't miss something. And so essentially what you're trying to do was balance precision and recall. You wanted to make sure you had high recall, where recall is, making sure that when something really is there, that you are scanning it. But I guess what I was asking was a little bit more about bias. Whether or not, even though the things that you're identifying is correct, whether or not you are systematically excluding a certain class of peptide identifications.
Yeah. I mean, so I reckon that could be, there is some potential for that, in a certain subset, but I think one thing we tried to lean towards in terms of this bias. So it comes back to how we do our offline analyses and our online analyses, right? It's, one of the reasons we stuck with Comet, right, this open source platform to do our online analyses was that we are literally doing the exact same scoring, ranking, and peptide spectral match assignment online that we do offline. So effectively, if we're biased, we're going to be biased online and offline, and that's just a general problem for these peptide spectral matching schemes, right? So that was a big push, and Steve was really excited about this, to really do the exact same scoring online and offline. So there's no potential bias. And I think you hint at something too that's an important thing to address too, which is like if you run different search algorithms, there is some slight bias just running different search algorithms across, even on the exact same sample, right? Well, it might not be huge. You might get a few hundred extra proteins out of a data set of thousands of proteins, right? That are just a little bit different. They were picked up by one algorithm and not another algorithm. But I think, yeah, like you're kind of hinting at, one of the important aspects of this for us is do the exact same online framework. So the same false discovery rate estimation, the same monoisotopic peak estimation, the same peptide spectral matching, all of it, this whole pipeline, we're going to try and condense that down from something that could run overnight to something that we need to run on the order of milliseconds.
Yeah, that makes sense. That you're essentially running the same algorithm, and so there may be bias in that algorithm, but that's not anything new that you've introduced. Okay, so then I guess let's talk about whether you were successful. And I guess there are two ways of measuring success, which is in terms of number of identifications and in terms of speed. So do you want to talk a little bit about that?
Yeah. I would also just throw in the caveat of, could we make this work in the first place? As a success. And we got it to work, which I think we were all just stoked about. And this also kind of builds off, I don't think I would do there's previous work by, like Derek Bailey too, to try and develop kind of similar frameworks for real-time searching. And I think the application to multiplex quant is where it really hit the ground running. But so in terms of success, I'll get back to the question. So speed-wise, yeah. So we had to go through a number of iterations. And again, Jimmy Ng was instrumental in this in terms of development of a pipeline that could work fast enough and also with an actual instrument in the field to do this. And so we tested this over and over and over again. This took multiple iterations to kind of drill down and figure out where our memory leaks were and all the issues that inherently built up. And success in terms of that was what we measured is actually if we just take an SPS MS3 based analysis, right? And we just look at the average time it takes generally to acquire an MS2 and then the difference in time it takes for when that MS3 finishes being acquired, right? Or starts being acquired. That's about 500 milliseconds. Right, so that's our operating time. That's the maximum operating time we essentially have. We have 500 milliseconds to do everything we need to do. And so when we re-optimize the search and re-optimize the search and re-optimize the search, what we were able to actually get that down to is if we're going to do a full yeast proteome analysis, we could actually do a search in 7 milliseconds. We can do the monoisotopic peak estimation in about 100 nanoseconds. We can do the false discovery rate estimation on the order of, depending on how big our training data set is, somewhere between 10 and 20 milliseconds. And so all that time combined for like this yeast data set, right, we can do that well under 50 milliseconds, which is 10% of the total time that we had in the first place. So I would say that was super successful. That was awesome to see the first time we got that going. And I think Jimmy and I were both really excited by that. And so even when we scaled that up, when we started looking at full human data sets, right? So much larger databases to do these peptide spectral matches with. We found that we could still do those searches in about 10 to 20 milliseconds. So still super fast. for doing this real time because we're still way under kind of that MS2 to MS3 based time difference. So I would say successful, check. The other thing we really were curious about, Red, is where can we push this to, right? So, okay, we're more efficient. We can make these calls online. We can do real-time searching, and it's great, and the algorithm works, and all right, that's fine and dandy, but how can we actually prove that this is useful for the community and should be implemented? And so what we did is we targeted kind of a standard analysis we would do in our lab, right, which is replicates, a biological replicate analysis of different cellular systems. In this case, just 3 different cell lines. We did this across an 11-plex, so 11 different biological replicates of 3 different cell lines. And so what we wanted to compare effectively was the state of the art, right, is this SPSMS3 based analysis. It's the most accurate, the most robust for quantitation. And we know it takes about 36 hours to acquire data using this method. So what we asked ourselves is if we're going to acquire somewhere in the order of 60 to 70% fewer MS3s, right, could we potentially reduce the acquisition time and get the same number of quantified proteins in the end? And so what we did is we actually just cut our instrument gradient time, so our acquisition time, in half. So a normal method set would run for 36 hours. With real-time search, we're going to run that for only 18 hours. And then we just did a comparison, right? We took all the data, both went through the exact same pipeline, all the exact same quantitative filters. And then we asked how many proteins and peptides we actually quantified in the end. We were actually able to observe that in the SPSMS3 based analysis, we could quantify just over 8,000 proteins in 36 hours across all 11 different samples, right? So about 3 hours per sample. And then using real-time search in this analysis allowed us to quantify the same number of proteins, or nearly so, in half the time. So 8,000 proteins with real-time search in about 18 hours. And so now we've doubled our acquisition efficiency, which means we have twice as much instrument time to play around with.
So did you continue to push that? Like, what happens if you run it for only 1 hour?
Yeah, that's a good question. So that's something we're trying to do right now. I think is the question is how far can we push that? Can we right now? Right, this whole pipeline is predicated on the idea again. Chatted briefly about it before. This offline fractionation idea of fractionating down to 12 fractions. And the question is, do we need to fractionate to a full 12? Could we get the same information from 10 fractions and do that with 2 fewer analytical runs in the mass spectrometer? Could we do it with 8, 6, 4? Right, and every step, maybe even if we had to do a slightly longer LCMS-MS gradient, that would be fine because we're still doing fewer fractions. And so this is still kind of an open question, and we're hoping to kind of see where the threshold is for, even if we set it at, we have to quantify at least 8,000 proteins, where is the threshold for how far we can push real-time search? But kind of the canon right now is somewhere between 16 to 18 hours for doing most of these analyses.
That's awesome. Yeah, that seems like a super compelling line of work, being able to run these expensive, experiments a lot faster. I imagine would push the field forward. If you could actually just sneak into somebody else's lab and run an experiment, courtesy of Devin Schweppe.
Yeah. Well, see, that's how we could do it, right? It's like, we'll just make them both real-time search and then it runs the same overall amount of time, but we both get something out of it.
Exactly. Right. Yeah. So you were saying that other people in the past have been less successful at the same general idea. What do you think the major breakthrough was that allowed this to work for you?
Yeah, I would add a small twist. I don't think they were necessarily less successful. I think it was the applications weren't as potentially beneficial as this MS3 based quantitation. And I think the main things for that is previous iterations of this were primarily focused on maybe we can identify a peptide and change the fragmentation scheme. And maybe that'll help us. But one of the issues there was it gave some more information, but it didn't robustly really improve the efficiency of these analyses. I think the striking thing and the most important thing for this current iteration of real-time search is really the most accurate way to do multiplex proteomics relied on this really time-expensive SPS-MS3. And that is this huge hurdle to get around, right? And so people sometimes just don't do the MS3. They'll just do high-res MS2 instead of that, which is much faster, but more essentially introduces more of this quantitative interference. So I think the big thing that lended to the success of this project and the proof of principle by Brian Erickson beforehand was this idea that we had an issue. We had a big problem that we were trying to address, which was SPS MS3 data collection is a pain in the butt, and we need to get around it. And we'd like to do fewer if we can get away with it. So how can we get away with it? And before there was not really that killer application for doing this, right? So I think that was the big boost that we gave to at least multiplex quantitative proteomics.
Sounds like a compelling reason, now people actually want to do it. So I guess as a final question, this was a system that was meant to be integrated into a mass spec. What do you see the prospects of this being the standard data collection method for MS3?
Yeah, I think more and more I hear people talking about using this. I think the big thing is that you kind of again hinted at is that this has been fully integrated now into like the latest set of thermo-based high-resolution mass spectrometers, the Orbitrap Eclipse series. And so I think that, right, in all bioinformatics pipelines, right, if you can get that integrated into commercial products, if you can get that out there to the general community for use rather than kind of just one-off lab products, I think that has been a huge boon for actually getting people not only interested in it, but using it. And I think I'm encouraged by folks sending emails to my lab and Steve's lab and asking about optimizing these methods and getting them up to speed and how they can use them in their labs. And I think that, daunting in terms of the number has been exciting in terms of people still being stoked about kind of using this tech.
That's awesome. That's really exciting to hear that not just people are using it on one-off, but it sounds like companies are adopting it in the pipelines they provide along with the tools. So thank you for being with us.
No problem. Thanks for having me.
Yeah, and like we mentioned earlier, you're currently looking for grad students and postdocs. And where is it they can go to apply?
They can head over to schweppelab.org. Schweppe is spelled like the ginger ale, Schweppes without the S. And then just lab.org. And come visit us or feel free to shoot me an email.
Awesome. Well, thank you again so much for joining us.
Thanks for having me. It's a pleasure.